Sarah is a chemical oceanographer and isotope geochemist interested in the cycling of major elements such as carbon and nitrogen in the marine environment, to understand the ocean's role in climate, past, present, and future. She grew up in Johannesburg, South Africa. She got her BSc honors degree at Harvard University and a master's and PhD at Princeton University. She's now currently at the University of Cape Town as a senior lecturer in the Department of Oceanography, where she has been since 2016. And she's the PI of the new Marine Biogeochemistry Lab in the Oceanography department there. And her research group focuses on questions of carbon and nutrient cycling, ocean productivity, oxygen dynamics, and climate in the Southern Ocean, Benguela upwelling system, and subtropical South Atlantic and Indian Oceans. And we're very much looking forward to her seminar today.
All right, wonderful. Well, thank you very much for the invitation. I cannot see any of the participants now, the way that my screen is set up. So if something goes wrong, please just interrupt me, or if you have questions or something. So yes, so the title that I gave Margit was A Tale of Two Upwellings. Contrasting nutrient dynamics may explain the comparable fertility of the northern and southern Benguela upwelling systems, but I'm actually going to focus today mostly on talking about the southern Benguela, in part because that's the region where my group has done the most work to date. It's the region that I'm most familiar with, but also to try and tell a more complete story that is hopefully of benefit to the RGNO school participants. And the other thing I should say is that, I'm talking about these data and the story today, but the work has been done by a large number of people, including students, both at the University of Cape Town and elsewhere. And, I'm very grateful for all their contributions. And it's also government agencies in South Africa and in Namibia that have allowed for some of this work to take place. And we wouldn't be able to do it without their cooperation. So I'm very grateful for that. Okay, so, whoops. Make sure I can go forward. Okay, so just to get us started, this is probably something that you are all familiar with, but nonetheless, I'm talking about a coastal upwelling system today, and in the southern hemisphere, you have southerly or southeasterly winds blowing parallel to the coast that result in Ekman transport, so the transport of the ocean surface layer being offshore, as a result of the Coriolis force, which is induced by the rotation of the Earth. And so as the surface layer moves offshore, it is replaced by cold, nutrient-rich waters that upwell to the surface. So I think we all know that, and we probably also all know that there are 4 major eastern boundary upwelling systems, often referred to as EBUS. Never mind. I can hear my email pinging and I wanted to turn it off, but it's okay. And those include the Peruvian, Californian, Northwest African, and of course our Benguela upwelling system. And these are regions that are characterized very near the coast by low sea surface temperatures due to the upwelling of deep waters, as well as high levels of productivity as phytoplankton grow in response to the input of nutrients. And you are also probably familiar with the fact that while these regions occupy less than 2% of the global ocean surface area, they're estimated to be responsible for about a fifth to a quarter of the world's marine fish catch. So there's plenty of motivation or plenty of reasons why we might want to study these regions. And in addition, those of us in Namibia and South Africa, the Benguela is right on our doorstep and is of high socioeconomic importance to our countries. Basically, it is the region between the tip of South Africa, so where the Agulhas retroflexion occurs, all the way to the Angola-Benguela front in the north. The system is particularly interesting in that it's bordered to the north and the south by warm water. And certainly in the southern Benguela, the fact that the Agulhas current, the warm Agulhas current, plays a big role in the Cape Basin region has implications for the physical dynamics in the southern Benguela. The system is divided by the Lüderitz upwelling cell into a northern and southern Benguela upwelling system. I'm going to refer to them as the N-bus and the S-bus. You probably are all familiar with this. And really, they are two very different systems. The Luderus upwelling cell acts as a barrier, a pretty effective barrier to water mass transport and the transport of nutrients in organisms. And there's some modeling and satellite-based studies that suggest the Benguela is the most productive of the 4 eastern boundary upwelling systems. This is not universally accepted, but nonetheless, it is a very productive, socioeconomically important system, with the World Bank estimating that about 10 million people rely on this system and that its fisheries are worth about $500 million annually. Okay, so what I'm showing here is both the northern and the southern Benguela. These are data all derived, I think, from satellite. And what you're looking at on the left is Ekman transport, the transport of the Ekman layer. And if the value is positive, it means that that is net transport offshore. So you've got upwelling. If the value is negative, it would mean downwelling. But the one thing to notice here is that there's no negative values. The middle panel is chlorophyll and the right-hand panel is net primary production, presumably derived via some algorithm from ocean color. And the different colored lines are just different seasons. So we won't worry about those for now. But what you can see from looking at this is that the transport in the southern Benguela versus the northern Benguela, they're very different. Upwelling intensity in the northern Benguela is significantly, maybe as much, depending on the metric you use, as an order of magnitude higher than in the southern Benguela. So theoretically, this means that the northern Benguela should be being supplied with far more upwelled nutrients than the southern Benguela. And yet, if we look at the chlorophyll and NPP, net primary production, both of which are metrics for the fertility of the system, the fertility is pretty similar in the north and the south. So it raises the question of how, if you have very different fluxes of nutrients, new nutrients into the system, can you support similar rates of productivity in both subsystems? And so that's sort of, this question frames a lot of what I'm going to talk about, focusing on the southern Benguela. But before I do that, most of what I'm going to talk about is related to the nitrogen cycle. I am, as Margit said, a chemical oceanographer, and I mostly measure nitrogen fluxes, nitrogen isotope ratios, because the nitrogen cycle is so useful for getting at all these different biological and chemical processes that are going on, and it's so intimately tied to carbon, which we'll talk about a little bit more. But just so we're all on the same page, I just wanted to go through the different biological processes that I'm going to rely on in my talk today. So the first is, I will use the words consumption and assimilation interchangeably. So the consumption of nitrate or the consumption of ammonium, for example, means nutrients, nitrate and ammonium both being nutrients, being taken up by phytoplankton in surface waters in order to fuel photosynthesis. And this nitrogen ends up being converted into PON, which is particulate organic nitrogen. So I will basically use PON and phytoplankton and biomass sort of interchangeably in this talk. The next process that I'm going to talk about a lot is remineralization or regeneration. I tend to use the two words interchangeably. And what this means is the conversion of the organic nitrogen bound up in particles in PON back to its inorganic form. So PON is first converted to ammonium, and then that ammonium is nitrified, oxidized via nitrite to nitrate. So remineralization is the process by which organic nitrogen in biomass is converted back to its nutrient form, so ultimately back to nitrate. And the step that takes, or the steps that take ammonium through nitrite to nitrate are termed nitrification. So I'm going to talk a lot about nitrification, but what I really mean is the regeneration, right? The recreation of nitrate in the water column. This is a process that requires oxygen and in net produces CO2, even though nitrification is actually a chemoautotrophic process. But the process of remineralization, converting organic nitrogen back to its inorganic forms, also converts inorganic carbon back to its, organic carbon back to its inorganic form, which is CO2. And then I will also talk about denitrification, which is the pathway by which nitrate is removed from marine systems. So nitrate is converted or reduced by various types of bacteria to ultimately to N2 gas. N2 gas is no longer available to organisms or to most organisms in the marine environment. Denitrification happens in the absence of oxygen, okay? And that's because these bacteria are basically using nitrate to breathe. They are replacing the oxygen with nitrate as an electron acceptor. And in so doing, they reduce the nitrate. And it's a process that produces CO2. But it's important to recognize that denitrification only happens at very low to zero oxygen. All right, so I said I was going to focus on the southern Benguela. So just this is the blue box here is showing you roughly the southern Benguela. This is a region of great economic importance to South Africa. It supports numerous commercial fisheries. The system itself is characterized by a fairly wide, shallow continental shelf. This becomes important. It experiences, this is a subsystem that experiences seasonal upwelling. So upwelling happens in the summertime when the southeasterly and southerly winds are blowing, and you tend to get quiescence in the wintertime. But even when there are periods of quiescence, which basically just means that you don't have upwelling favorable winds blowing, you don't get significant amounts of downwelling. So the net Ekman transport is always offshore, which means that even when you're not actively upwelling new nutrients to the surface, the fact that you've got this shallowing of the continental shelf means that the water mass isopycnals tilt up towards the surface, and you actually end up still bringing deep nutrients to the surface. It's just not happening in the sort of vigorous way it happens during active upwelling. And the other thing that's true of the southern Benguela is that it experiences frequent events of hypoxia, which is when the deep waters, so bottom waters on the shelf, basically run out of oxygen or run out of enough oxygen for lots of organisms to live comfortably. So in South Africa, we're pretty familiar with hearing about mass lobster walkouts, for example, off our west coast. And that happens because you get oxygen, these organisms become oxygen starved because the bottom waters become so low in oxygen. And so they walk out of the ocean. Importantly, in the southern Benguela, the low oxygen events are created only as a result of the biological or bacterial decomposition of organic matter. The source water, and I'll show some of this, but the source water that comes onto the shelf is well oxygenated. And this is in contrast to the northern Benguela, where there are periods where you get low oxygen waters coming onto the shelf. That doesn't happen in the southern Benguela. The removal of oxygen is purely driven by biology. I'll talk more about that. All right, and so the other thing that's important about the southern Benguela, or that's notable about the southern Benguela, is that on the continental shelf, it is characterized by a number of equatorward flowing hydrographic fronts. So what does that mean? Well, a hydrographic front is like a jet that's flowing towards the equator that gets set up because of a gradient in sea surface temperature. And so in the southern Benguela, you've got the Benguela current actually exists as both a coastal and an oceanic current. And the coastal current, when it comes around Cape Point and gets, encounters the continental shelf, it can split into what is often termed the upwelling front that I'll actually call the Columbine front, but that's the most inshore front. And that basically denotes the point inshore of that front, you get the upwelling of deep waters, and that sets up that front. So it's very close to the coast. And then at the shelf break, so where the continental shelf is about 300 meters deep, you get the setup of a second front, which is termed the shelf break front. And you also get upwelling happening just inshore of that front. And then finally, way offshore, you get further off the coast, you get the oceanic front, which basically represents the eastern boundary of the subtropical gyre. But what's important about these fronts is that they are strong and they act as boundaries to offshore transport. So they act as boundaries to the offshore transport of organisms, particles, things like that. And interestingly, I mean, they are surface features, right? These fronts are set up because of differences in sea surface temperature, but they nonetheless have an effect on the residence time of both surface and subsurface waters. And I'll talk more about how that works. And then depending on the strength of upwelling, you can actually get the fronts merging. So they're quite dynamic, they move quite a lot. So for example, in the wintertime, where we don't have very active upwelling, the offshore waters start to cool down and the inshore waters actually warm up because you don't have active upwelling. You end up eroding some of the sea surface temperature differences and the fronts will merge further offshore. And this is going to become important, this movement of the fronts. And I just like to show this plot from some of Taryn Lamont's work. And what you're looking at here is temperature, conservative temperature on the y-axis and salinity on the x-axis. And it's just showing you a number of different stations sampled from the inshore to the offshore of the southern Benguela. Thank you. So in the summer, where you have active upwelling, you can see that the inshore waters cluster very closely together, and they're separated from the midshore that then clusters together and further separated from the offshore. And so these inshore waters would be inshore of the upwelling front. The midshore would be here between the upwelling and the shelf break fronts, and the offshore waters then over here, right? They're very clearly separated. Whereas in the wintertime, you get a lot more sort of merging of these waters because the fronts are less strong, the temperature gradients are less strong. Okay, so we've done a lot of work in the southern Benguela. The South African Department of Forestry, Fisheries and Environment, they run regular quarterly hydrographic cruises or monitoring cruises in this region, and they sample all 4 of the monitoring lines that you see on this map. And so today I'm going to show data just from what I'm calling summer and winter. It's really February and May. May is kind of early winter, but February represents towards the end of the upwelling period, and May represents a period of quiescence. And you can see from this map, the shading is sea surface temperature. And in the summertime, we had all 3 of the major fronts present, right? The Columbine or upwelling front, shelf break front, and oceanic front. Whereas in the wintertime, the Columbine front and shelf break front had merged. And so we only had the 2 fronts. So we have a lot more data than just these 2 seasons, but I'm just going to show these seasons for a point of comparison. Okay, so I'm going to show a lot of plots that look like this, section plots across the system. And I'll typically show them from just one of the monitoring lines, but for the most part, not always, but for the most part, the trends that we see along one of the lines, we see along all of the lines. It's pretty consistent, except the southernmost line, where the continental shelf is very narrow, but that's sort of beyond the scope here. All right, so what you're looking at here is the colors are density, and I'll start at the top. This is the summer, and they're just 2 of the different monitoring lines. And you can see this yellow colored water mass that's outcropping right at the inshore here. This is subantarctic mode water. Subantarctic mode water is the sort of dominant upwelling water mass in the system. It's overlaid by South Atlantic subtropical mode water, which in the summer upwells offshore of subantarctic mode water. And in the winter, which are the panels at the bottom, tends to be the only upwelling water mass. You can see that in the winter, subantarctic mode water doesn't make it all the way to the surface, but the South Atlantic subtropical mode water does. Some of you might be more familiar with South Atlantic central water, or as it's sometimes referred to, and South Atlantic central water in this case would be a combination, not an exhaustive combination, but a combination of these 2 water masses, but it's actually useful for our purposes to distinguish between subantarctic mode water and South Atlantic subtropical mode water. And South Atlantic subtropical mode water actually derives from subantarctic mode water anyway. So subantarctic mode water is the ultimate source. It's to the system, it's the nutrient conduit. It's what supplies the nutrients. Okay. Oh, and the last thing I should have mentioned, I beg your pardon, is that I always indicate the positions of the fronts by these triangles at the tops of the plot. And there's always a little legend on the side. The names of the fronts are less important than the fact that between summer and winter, they move, they merge, and you also end up with fewer of the fronts being present in the wintertime. All right, so let's look at some of the nutrients. In this case, I'm actually showing all the monitoring lines. And what you are looking at are the nutrient concentrations in summer, nitrate at the top, phosphate at the bottom. And really the thing to take away from this, if we look, for example, at this Kleinseer monitoring line, nitrate concentration over here in the top left-hand corner, what you can see, the contours are still density. So the nitrate concentrations on the shelf, the phosphate concentrations on the shelf, inshore of the Columbine front, and to some extent inshore of the shelf break front, are higher than the nutrient concentrations in subantarctic mode water offshore, which is kind of curious, right? So you've got this upwelling source water that has a nitrate concentration of around 20 micromolar, and we sometimes measure concentrations as high as 35 micromolar nitrate on the shelf. So the on-shelf nutrient concentrations are higher than in the source subantarctic mode water. And if we look at the winter, so this is exactly the same setup, but now for winter, we see the same thing, but now the elevated on-shelf nutrients seem to extend further offshore. So you can see where the nitrate was sort of retained here. In the summertime, it now extends further offshore. The same is true for phosphate. So one of the things that we do is to calculate a delta nitrate or delta phosphate. All that is looking at the measured nitrate concentration minus the source value. So for both nitrate and phosphate. And when we do that, that's what's plotted over here for summer on the left, winter on the right, nitrate at the top, phosphate at the bottom. What you can see is that accumulation, or what we interpret as an accumulation of nutrients in excess of what's supplied by subantarctic mode water. And that gets higher than 10 micromolar nitrate and 1.5 micromolar phosphate, which is not insignificant. That's a lot of nutrients that are there on the shelf that are higher than could have been supplied by subantarctic mode water. And again, you can see that this, what you might call a bolus or a pocket or pool of excess nutrients, extends further offshore in the wintertime than in the summertime. Okay, so our hypothesis is that those nutrients come from remineralization that's happening on the shelf. So remember, remineralization is the process by which organic matter is converted back to its inorganic forms. So on this little schematic that's represented here at the bottom, so you've got an organic molecule or organic matter represented by CNP. And in the presence of oxygen by bacteria, this organic matter is decomposed into CO2, nitrate, and phosphate. So what you're seeing at the top here is simply the process of photosynthesis, right? Where CO2 and nutrients are used up to create that organic matter in the first place. So we hypothesize that the excess nutrients on the shelf come from organic matter created in the overlying surface waters that sinks onto the shelf and is remineralized back into the nutrient forms. And if that's true, there should be a correlation between the nutrients on the shelf, the excess nutrients on the shelf, and the amount of oxygen on the shelf, because to decompose or to break down organic matter requires you to use up oxygen. Okay, but instead of looking just at oxygen, the oxygen concentration, because oxygen is affected by things like exchange with the atmosphere as well as by biology, we want to look at just the component of the oxygen that is influenced by biological processes. So we define, or we use something called apparent oxygen utilization, which is what it sounds like. It's how much oxygen was used by biology. Okay, and all that is the observed oxygen concentration minus the theoretical oxygen concentration if the oxygen concentration were due only to physical processes, so to like exchange with the atmosphere. So in other words, we're just removing the physical component and we now have the biological component left behind. So AOU, the higher the AOU value, the more oxygen has been used up, right? It measures the amount of oxygen consumed by respiration. And so we would expect a relationship between AOU and carbon, nitrogen, and phosphorus. For those, many of you will be familiar with the Redfield ratio, but for those who aren't, carbon, nitrogen, and phosphorus, when these organic components are broken down, they tend to exist in the marine environment in a molar ratio of 106 to 16 to 1. So to break down that, you would require around 138 moles of oxygen. So AOU would change by 138. This value is variable, been shown to be variable depending where you are in the ocean, but we use 138. Okay, so the whole point is if the nutrients are coming from regeneration on the shelf, as opposed to, for example, being transported from the north or transported from the south, you would expect a relationship between the excess nutrients and AOU, and that's exactly what we see. So here I'm showing phosphate on the shelf in summer and in winter, and you can see that the distribution of the excess phosphate aligns beautifully with where we have high values of AOU, in other words, high amounts of oxygen that have been consumed on the shelf. So all of this suggests very strongly that these nutrients are being regenerated in situ on the shelf. And similarly, if I just look at a different monitoring line, just to show that this signal is pervasive across the southern Benguela, you can see again that where we have high phosphate accumulating inshore of the Columbine front over here, we have very high AOU. And similarly in winter, we see the signal, but now it's more spread out on the shelf. But it overlaps with the AOU. Okay, so the idea here is that the Southern Benguela upwelling system facilitates something called nutrient trapping. So we did not propose the idea of nutrient trapping. This is something that was first put forward for the Benguela as a whole by Tyrell and Lucas in a 2002 paper. I think I gave this paper to the students if they were interested. And the idea is that you have subantarctic mode water, it upwells onto the shelf, okay, ignore the regenerated nutrients for now. It upwells onto the shelf to support productivity. The productivity in surface waters creates large amounts of organic matter. They sink to the shelf bottom and get regenerated and create this big pool of regenerated nutrients on the shelf. On the right-hand side here, you're just seeing our measurements, which correlate exactly with this cartoon. And then the idea is the next time you upwell subantarctic mode water, you augment its nutrient concentrations with these additional nutrients on the shelf. So in other words, you raise the concentration of subantarctic mode water and supply more nutrients to the surface to support productivity. And so the other part of this that is key is that at the same time, so you've exported the organic matter, remineralized it on the shelf, you continue to advect these now nutrient poor surface waters, off the shelf, right? Ekman transport of surface waters off the shelf. So you've taken nutrients, trapped them on the shelf, and taken nutrient poor waters and sent it back off the shelf. And in augmenting the nutrient concentration in subantarctic mode water, you can now support higher rates of productivity. And it's possible that this is what explains why the Southern Benguela is as productive as the Northern Benguela, even though the total amount of subantarctic mode water, so like pure subantarctic mode water that comes onto the shelf, or the total amount of upwelling of that subantarctic mode water is a lot lower in the Southern Benguela than upwelling to the north. But what the work that we've done so far adds to this argument is that these hydrographic fronts, which you're seeing here represented by stream flows going into my page and the fronts by triangles, the presence of these hydrographic fronts is what helps to trap the nutrients, because the hydrographic fronts prevent the biomass that's created in these surface waters from advecting offshore with the surface waters. So the biomass is retained inshore of the front, so that it can sink and be remineralized on the shelf. Similarly, between, say, the upwelling and the shelf break front, we see the same thing. So you should see sort of two blobs of regenerated nutrients between the fronts. And this explains, or can explain at least, why the elevated nutrient pool extends further westward or further towards the offshore in the winter when the fronts move further to the offshore. And so the idea here is that nutrient trapping is aided by the presence of these equatorward flowing fronts. Okay, so one of the things that you might want to do, because now we're saying that trapping these nutrients on the shelf enhances the amount of productivity that can happen in the Southern Benguela. So it might be useful to be able to quantify the amount of nutrients that you're actually regenerating on the shelf. So what I'm showing here is just the phosphate concentration on this plotted against AOU and the nitrate concentration plotted against AOU. So I told you before that we expect these molar relationships between phosphate and AOU, right? You would expect a ratio between phosphate and AOU of 1 to 138, and that's what this dashed line is showing. And if everything in the system was behaving according to Redfieldian expectations, then that's exactly what you would see. All the data would fall on this line, on this slope. The open black symbol is subantarctic mode water. So this is our starting point, right? But in fact, what you see is that there's decoupling, and it's even more evident for nitrate than it is for phosphate, where the data points fall off the line. So if we wanted to use the measurement of AOU, to try and determine the amount of nutrient that's been regenerated on the shelf, we can't do that because there's a decoupling between the nitrate and the phosphate in the system. And this happens because phosphate can be adsorbed onto sediments or released from sediments independent of nitrogen and, very complicatedly related to oxygen conditions. And nitrogen can be lost in the sediments, or nitrate can be lost in the sediments to denitrification, but the nitrate that's in the waters just overlying the sediments is what actually gets into the sediments to fuel denitrification. So you can lose nitrate. And that seems to be true. We have a big nitrate deficit on the shelf, even though we still have excess nutrients. Okay, so we can't use sort of the standard, what is a standard chemical oceanography approach to calculate the burden of excess nutrients. So we turn to the isotopes of nitrate, and this is what my group does a lot of work on. So nitrate is a molecule with one N atom and 3 O's. It's very useful because we can measure both the nitrogen and oxygen isotope ratios. Here I'm talking about the natural abundance ratios. And the idea is that nitrogen exists mostly as N14 with a small amount as N15, and the ratio of the N15 to N14 in a particular sample, so nitrate dissolved in seawater, for example, is set by the different biological processes acting on the nitrate pool, as well as by, you know, physical processes like mixing. And so we can use the nitrogen isotopes to track physical and biological transformation. So hopefully that'll become a bit clearer as I go through this. And importantly, on the left, I'm showing nitrate consumption by phytoplankton with the nitrogen, the delta O18, so just the oxygen isotope ratio of nitrate on the y-axis, the nitrogen isotope ratio on the x-axis. The fact that these are isotopes is not particularly important. What is important is that when phytoplankton consume nitrate, the oxygen isotopes and nitrogen isotopes evolve in a ratio of 1:1. This is kind of remarkable, and I don't have time to talk about why this is, but it's true. So phytoplankton nitrate assimilation or consumption creates a very obvious pattern in the nitrate pool of this change in a ratio of 1:1 between delta O18 and N15. By contrast, something like nitrification, so, you know, we're interested in regeneration on the shelf. Nitrification is part of that process. What it does is it does not change the delta N15 of the nitrate pool, but it lowers the delta O18. Okay, so this is really the only thing you need to take away from this. If we have nitrate consumption, we increase N15 and O18. And if we have nitrification, we don't increase N15, but we decrease the O18. Okay, and the other thing is denitrification, the process that removes nitrate from the system, has the same effect as assimilation. So it also raises N15 and O18. All right, so if I've completely left you behind, not to worry, I will explain how this applies to what we're looking at. So we measure the nitrate isotope ratios in the system, delta N15 and delta O18. You're looking at N15 at the top for summer and winter, O18 at the bottom for summer and winter. And the first thing, or the only thing I want you to take from this slide, is that the delta O18 of nitrate is higher on the shelf than in subantarctic mode water. Okay, that's particularly obvious in winter. You have this much higher delta O18 on delta N15, sorry, on the shelf than in subantarctic mode water. And the delta O18 is lower on the shelf than in subantarctic mode water. So why is this? What are we actually seeing? Okay, let's zoom in on the delta O18 first. So the delta O18 on average on the shelf, where we have that pool of regenerated nutrients, ranges from about 1.8 to 2.6 per mil, whereas the source subantarctic mode water has a delta O18 of more like 3 per mil. So this is all, remember I said nitrification decreases the delta O18. So this is all consistent with nutrients being added by nitrification, by regeneration on the shelf. And if we look at the delta O18 of that nitrate plotted against oxygen again, remember regeneration decreases the oxygen concentration, so increases the AOU. What you're looking at here in the gray line is the delta O18 of subantarctic mode water. On the left here, where the delta O18 values get very high, that's photosynthetic assimilation, right? That's phytoplankton consuming nitrate in the surface. And then here, where you see the data all going below the subantarctic mode water line, the delta O18 gets very low, that's regeneration. Okay, you've got nitrification on the shelf, which is decreasing the delta O18 of the nitrate in both summer and in winter. And then interestingly, particularly in winter, at the highest AOU, so the very lowest oxygen concentrations, we actually see a rise again in the O18, and that is indicative of water column denitrification, which is generally thought not to happen much in the southern Benguela. And this is only actually in St. Helena Bay, for those of you who are familiar with the system, but it does seem that there are pockets of water column denitrification happening here. Okay, so the reason I'm telling you all of this is to convince you that the O18 is reflecting remineralization on the shelf. And if that's true, we can use that fact to calculate the amount of nitrate that was regenerated on the shelf. Because we assume that the nitrate that we measure on the shelf, that its delta O18 is set by the delta O18 of subantarctic mode water and the delta O18 of what was remineralized, right? Mixed together, you get what you observe. And so when we do that, we can solve for F, which is the fraction of the nutrients that are regenerated. And what we calculate is that roughly a third of the on-shelf nitrate is regenerated in summer. That means about 10 micromolar, and as much as 45%, so more like 15 or so micromolar in the wintertime. So that's a lot of nitrate being regenerated in situ on the shelf that is then available to be upwelled to support productivity. If we look at the delta N15 of nitrate, as I said before, it's higher, it rises on the shelf relative to subantarctic mode water. So it's about 7 to 8 per mil versus 6.3 to 6.5 in subantarctic mode water. That's particularly evident in winter. And that is indicative of sedimentary denitrification. So I alluded to this before, where the nutrient ratios become decoupled. But basically what's happening is you're regenerating a huge amount of nitrate on the shelf, but at the same time, some of it's being used up for denitrification in the sediments. And I think I'll just leave this out, but what that means is that more than the amount of nitrate that we calculated is actually regenerated on the shelf. And so that means that our calculations of the fraction of regenerated nutrients is actually a lower bound on how much is being regenerated on the shelf. So it's a lot of the nutrient pool. So this is clearly a very regenerative system. And that is in direct comparison, for those of you familiar with the Northern Benguela, to what is the Northern Benguela, has very high levels of new production, so new nutrient-supported productivity. All right, so if I just take a step back and summarize what we think the nitrogen cycle looks like on the Southern Benguela shelf. Here I'm showing delta O18 as a function of oxygen now, no longer AOU, but oxygen, and delta N15 as well as a function of oxygen. Okay, subantarctic mode water, so the starting value is indicated by the black dot. So the first process is photosynthesis that's happening in surface waters. And what you see there is that the delta N15 and the delta O18, sorry, the other way around, O18 and N15 of the nitrate rise by the same amount due to photosynthesis, okay? And oxygen is produced during that process, right? The oxygen concentrations rise. On the shelf, we see remineralization happening, so nitrification, which decreases the delta O18 and increases the delta N15 because you've got sedimentary denitrification happening at the same time. And then we also see these incidences at very low oxygen of water column denitrification, where both the delta N15 and O18 rise again. So hopefully this gives you a sense of how powerful the nitrate isotopes can be coupled with the nutrient data to give us a sense of the different overlapping processes that are going on the shelf. So I'm just going to talk for a couple of minutes about the implications of this. And the major implication is, of course, about oxygen. Right, the more remineralization that you have on the shelf, the more oxygen you're going to strip out of the bottom waters. And so one of the things that we are now trying to understand with the hope eventually of having a predictive understanding so that you could sort of maybe predict what the oxygen conditions might be like based on, the variety of meteorological and physical conditions. So when you have what you might consider normal upwelling, so summertime upwelling, right, you have all three of these fronts set up on the shelf. You have really intense upwelling inshore of the upwelling front, so right near the coast, and you would accumulate nutrients on the shelf that lead to hypoxic waters right inshore, okay? When you have a scenario where the columbine front or the upwelling front collapses onto the shelf break front, and this can happen when you have quiescence, like I showed for winter, but it can also happen when you have extremely vigorous upwelling, so that you're bringing so much cold water to the surface that it actually erodes the sea surface temperature gradient and forces it further offshore. Then you're going to have a larger area over which you regenerate nutrients and thus create hypoxia. And then finally, you've got the option where you have a weak upwelling front, and very small amounts of nutrients coming to the surface so that you will have lower nutrient trapping and less hypoxia. This seems like a more idealized idea to me. But in any case, And the thing that we're currently missing in terms of our understanding of the system is you can't accumulate nutrients and lose oxygen on the shelf forever, right? You have to flush the shelf at some point because we don't see these nutrients go up and up and up year upon year. And that's probably happening at the onset of upwelling. But one of the things that we're trying to better understand is what that looks like physically and what the implications of that might be. Another implication, and I'm just going to mention this very briefly, is about the production of nitrous oxide gas. So this is something that I'm starting to think about more and more in this system. Nitrous oxide is a very powerful greenhouse gas. On a per molecule basis, it's about 300 times more powerful, or has 300 times higher radiative potential than CO2, so very strong greenhouse gas. Its concentration in the atmosphere is rising due to anthropogenic activities. And the ocean is actually a fairly significant source, in this case, a natural source, but a fairly significant source of N2O to the atmosphere. It's about 20% of the global N2O sources. And of the ocean source, the eastern boundary upwelling systems account for about a fifth to maybe a quarter of that. They are hotspots for N2O production. Okay, and that's what this map is showing you, is the N2O flux to the atmosphere. And while the Benguela is not particularly well resolved, I mean, I guess you can see part of the northern Benguela, the eastern tropical North and South Pacific, where you have the upwellings, you can see these really high fluxes of N2O to the atmosphere. N2O is produced under conditions of low oxygen by nitrification and denitrification. So when nitrifiers become stressed because there's low oxygen, they can produce N2O. And similarly, when denitrifiers do not have enough, well, when denitrifiers have too much oxygen that they can't reduce nitrate all the way to N2 gas, they can often stop at N2O and produce N2O. So low oxygen conditions are perfect for creating high concentrations of N2O. And then when you upwell, you then supply this powerful greenhouse gas to the atmosphere. And so this is a really important implication of regenerating a lot of nutrients in an upwelling system. And so what I'm showing here are just some measurements of N2O in the southern Benguela, just to show you that this correlation with oxygen is a reasonable thing, you know, it's a reasonable implication of the work we've been doing. So at the top here, you're looking at AOU, and at the bottom, delta N2O, it's just the amount of N2O in excess of the saturation value. So it's the amount of biological N2O. And you see where you have a lot of AOU, so low oxygen concentrations, because you've had a lot of oxygen consumption, you get high concentrations of AOU, right? And it seems likely that the same idea that the fronts are going to affect the extent to which you end up generating these high concentrations of N2O on the shelf. But this is pretty much a work in progress, so that's all I'm going to say. But we're also using the isotopes of the N2O, including the isotopomers, for those of you who have a sense of that, to deduce what processes are resulting in the production of that N2O. Okay, so to conclude for the southern Benguela, our hypothesis is that the remineralization that happens on the shelf exerts a positive feedback on the productivity of the system. So in other words, you enhance the subsurface nutrient burden that is available to be upwelled. So in subantarctic mode, water upwells again, it can entrain these high concentrations of regenerated nutrients and support a lot more productivity than it would otherwise be able to. We're hypothesizing that the hydrographic fronts play an important role in the trapping of nutrients on the shelf, and that this has implications for hypoxia and N2O production that we'd like to understand better, particularly as upwelling changes, right? What happens if upwelling intensifies? Do you move the fronts further offshore and thus have a longer residence time for inshore surface waters, more time to accumulate biomass, more organic matter going to the shelf, and more oxygen being stripped out? Or does more vigorous upwelling mean you have phytoplankton actually have less time to consume the available nutrients before those water masses are advected offshore? We don't know the answer to these questions yet, but these are some of the things that we're thinking about. And then I had a couple of slides about the northern Benguela, but I think I'm not going to show them, and instead I'm just going to say that what's different about the northern Benguela system is that the continental shelf is a bit narrower, and it doesn't have the same hydrographic fronts as in the southern Benguela. So there's less, if we are correct about the hydrographic fronts contributing to nutrient trapping, it's likely that there will be less intense nutrient trapping in the northern Benguela. And it's also likely that there will be more intense denitrification in the northern Benguela because it has periods where it goes anoxic, which would facilitate water column denitrification. So it's possible that the combination of not as much nutrient trapping or not as intense nutrient trapping, coupled with the fact that you actually lose quite a lot more nitrate to denitrification, means that you don't have the same amount of regenerated nutrients to support productivity in that system. So it's a much more new nutrient supported system than the southern Benguela. And of course, even though the productivity of these two subsystems seems to be roughly the same, the fact that one is supported by a lot of regenerated nutrients and the other mostly by new nutrients has really important implications for things like carbon export, right? Because when you regenerate nutrients, you regenerate CO2 as well. And when you bring those nutrients to the surface to support productivity, you ventilate CO2. So it doesn't mean that the southern Benguela is as good at drawing down CO2 as the northern Benguela. And I think, so this is our working hypothesis about those two subsystems. I think I will just take a second to acknowledge the myriad of people who have contributed to making this possible and to whom I'm very grateful. This is very much a work in progress still. And then I think I'll just leave this slide with our kind of working hypothesis, and then I'm happy to take any questions or talk about any aspects of this that you would like to talk about. No problem.
I see Volker has a question.
Yeah, that's true. First, Sarah, thank you very much for this really excellent talk about the southern Benguela, and I think I learned a lot because I just work normally mostly in the northern Benguela, but I'm not quite sure whether, or I have some doubts about all this frontal stuff, because if you discuss 3 different fronts, I'm not quite sure whether they are really there or whether it is a little bit a bias of the high mesoscale variability we find in the area, especially in the southern Benguela, where the mesoscale activity or features are much stronger than in the northern one. And the thing is, we have run for several years hydrodynamic models, and these models doesn't really will support the existing of such pronounced or distinct fronts in the entire Benguela area. There are always fronts like from filaments, from smaller eddies, or like, things like that. Also, if just shortly after the onset of upwelling, we have distinct temperature fronts in the near the coast. But often we see, for example, at the position of this shelf break front, these piling up of thermoclines we often see there is not really a real front. It is driven by turbulent processes like breaking internal waves at the shelf edge or so. And there is not really up or down welling. It is just in vertical mixing of water columns. So, and therefore, I totally agree with this story of trapping nutrients on the shelf that we see also in the northern Benguela quite well. But I'm a little bit in doubt whether that will be supported by these type of fronts you have mentioned.
Yeah, well, thanks for the question. So, I mean, you make a really good point, right? At the point of doing the biogeochemical component of the study, it really just was a hypothesis. And it's mostly because we see such a strong relationship, and admittedly it's qualitative, but between the position and the extension of these regenerated nutrient pools and the positions of the fronts. And people have seen this previously with things like oxygen on the shelf. But in fact, one of the things that I have a PhD student now working on pretty intensively with Jenny Veach at SEON, who is a physical oceanographic modeler, is the development. So we're using, he's been working on a 3-kilometer ROMS simulation to see whether or not we get particle retention once we are happy that we have the physics right, which we've pretty much got to, whether or not, you know, it does seem like these fronts are what is leading to the nutrient trapping. And indeed, I mean, he does get in the 3-kilometer resolution, the fronts being set up in the model and being coincident with appropriate vertical velocities. But I agree with you that I have given you a very sort of singularly dimensional view of what's going on, and the fronts, that's not what they look like. But yeah, this is something that we're definitely working to try and understand a lot better, and the physics are absolutely paramount in doing that. But it is true that the Benguela jet, that is a feature, that is a real feature that people have identified, and it shows up, in these southern Benguela models. So I think there's something in it. We'll see. But yeah, I take your point. Should I answer Jake's question that's in the chat? So, are you aware of the Kraft 2022 paper reporting oxygen production by ammonia oxidizing archaea? Could that be relevant to the Benguela? Yeah, so that sort of blows my mind about how the nitrogen cycle is working and what the implications of that might be. I'm sure it's relevant in the Benguela. There are currently only a handful of unpublished measurements of nitrification direct measurements of nitrification in the system, and no work on AOA, on ammonia oxidizing archaea at all. But I suppose it's possible that the effect of that, the production of nitrate and coincident production of oxygen has implications. I don't know. It's definitely something for us to think about. The relative import of that flux of oxygen compared to how much is being removed, that I don't know. That's what it will come down to, is the relative magnitude of those fluxes. The other thing that's lacking in the system that would be really useful to do is is some work at the sediment water interface. And that's not my wheelhouse at all, but with N2O production as well, we don't know how much of that N2O is potentially being produced in the sediments or being consumed in the sediments relative to what's actually happening in the water column.
Great, thank you.
Good.
Yeah, thank you, Sarah. It's a lot of food for our course. And luckily we have a means then, if we have it recorded, to listen to that recording at half speed. For us, that's always a problem for us. Follow it. But let me go to the facts. Well, there's little oxygen, little nitrate, and those two together should take care of the remineralization of all the organic biomass that had been produced in the surface waters. That's stoichiometrically just not possible, as you point out too. Why is the sulfate always left out of these considerations? Because there is so much more sulfate than any of the other oxygens.
Right, so in the southern Benguela, at least do you mean in the water column or in the sediments?
In the water column.
Okay, so in the southern Benguela, for the most part, the oxygen concentrations do not get low enough. We have pretty good handle on the annual or monthly climatology of the oxygen concentrations from the deployment of moorings by the South African groups, the German groups, etc. So the oxygen concentrations don't really ever get low enough that water column denitrification even becomes that much of a thing. It seems like there are maybe little pockets of it. So, I guess the sulfate would then be the next most advantageous electron acceptor after the nitrate. But I believe in the northern Benguela, the sulfate in the water column you definitely get hydrogen sulfide production, right, from the reduction of sulfate. I'm not super familiar with that. But I think in this system, that's the reason, is because at least in the water column, the oxygen is generally just not low enough. And that's sort of what I meant by saying, we see oxygen being stripped out of the water by this regeneration, but at some point in the annual cycle, it is being flushed and replenished enough that we're not, driving it to complete anoxia. And part of the reason why that's easier in the south than the north is because we have the oxygen, the water mass that's sloshing onto the shelf is oxygenated subantarctic mode water. I mean, the oxygen concentration is over 200 micromolar. Versus
I fully agree. That would also mean, however, that a lot of the biomass reaches the bottom without being degraded or partially degraded. So whatever happens in the water column with regard to regeneration of nutrients is, let me put it a little provocatively, it's minimal compared to the nutrients that get trapped in the sediments. Is that correct?
Possibly, yes. And that's an almost impossible thing to ever quantify, because the nutrients that are trapped in the sediments, I mean, are being denitrified. The phosphate is, all sorts of things are happening to that. I don't know that it's trivial though. Right.
Well,
I mean,
do you know anything about how much of the primary productivity is actually reaching the bottom in terms of carbon or nitrogen?
Yeah, it's a really good question. I mean, so there's not very many direct measurements of that flux. There are proxy measurements, but those, of course, suggest that in the southern Benguela is surprisingly regenerative, as opposed to, you know, but in terms of like actual measurements of direct measurements of the carbon and nitrogen flux making it to the shelf bottom, I don't know.
Mm-hmm. Mm-hmm.
I think a lot of it is regenerated.
Well,
but it would be interesting to do that back of the envelope calculation, right?
Well, I might disagree on that one, but it's degraded, you know, considering how rich the sediments are with regard to organics. But Margaret, if I may ask another question, or you have someone in the chat?
Go ahead, good.
Okay. We learned last Thursday from Olivia things that Jake has mentioned already, but also that there are organisms that can degrade ammonia with oxygen. And I'm wondering, have you ever considered or found any evidence that ammonia can also be oxidized by nitrate? Anammox process?
Yeah. No, but that doesn't mean it's not happening. So, you know, when I talk about there being some evidence of water column denitrification in the nitrate isotopes, that means denitrification in the very general sense that it probably includes anammox, because the empirical work that's been done to quantify the effect of denitrification on the isotope ratios, a lot of that does not distinguish between, you know, canonical denitrification and anammox. So I have no sense of how they separate out, how important one is relative to the other, but it's certainly plausible, you know, if denitrification is happening and you have all these electron donors and acceptors, that if that were energetically favorable, I would imagine that, yes, it's happening. And you can have fairly significant ammonium accumulation, as you might imagine, in the system. If you are getting all this regeneration, you know, sometimes we see ammonium concentrations of 6 or 7 micromolar. You would think that somebody's metabolism would have adapted to be able to exploit that. But yeah, there's been very little sort of molecular level work done in this system to get at things like that. Mm-hmm. But yes, when I say denitrification, I mean sort of the removal of nitrate and its conversion to N2, sorry.
Well, that's my other point. You say N2O, sorry, N2, or the vice versa. Why is it not converted to N2 completely? I mean, it would be an additional two electrons per N2 that could be deposited. Right. Why is that not being done?
So, I mean, it is, but so the enzyme that catalyzes the final step in that pathway is very easily poisoned by oxygen. Like, it's super oxygen sensitive. So if you have in the southern Benguela, you know, the oxygen concentrations, it gets hypoxic, but it doesn't get below well, it doesn't get perennially or broadly, it can happen in pockets below about 40 micromolar or so. And that's still high for denitrifiers. And so it's the reason you would they stop sometimes at N2O is because the oxygen concentrations are actually too high for the final step, or at least that's what the community at large thinks is the case. But eventually, you know, the N2O will be consumed. I mean, denitrification is the only way to remove N2O unless you flux it out to the atmosphere from a system. So eventually it will happen. You're right. But it's not just a consideration of the energy, it's also the oxygen.
Thank you very much.
Sure, no problem.
I haven't seen any other hands up. Are there any questions? Yeah.
From the students?
From any students online or in person?
In the lecture room?
But I don't see any hands. Thank you so much, Sarah, for
no problem.
This very interesting talk. I certainly know a lot more now about that. And you can always go back to the video and watch it again.
Yes, I'm sorry if it was that it might have been too fast. The one thing I will say about that is that this is a very classic application of chemical oceanography.
Yeah.
And I think that it's so elegant for that reason, right? That you can look at these large scale stoichiometries between the nutrients and oxygen and deduce, and it's really the deviations that tell you a lot, but deduce some really interesting things about what's going on. So apologies if I spoke too fast, but I did give Kurt a couple of papers that if you're interested, if the students are interested, will hopefully be helpful in sort of making some of it a little clearer.
Yeah, the papers are available online under your name on the program. So thank you very much for sending me those. Okay? But we have now again food for the course.